warahmatullahi wabarakatuh. Hi everyone. I will discuss with you chapter 1 on topic Introduction of Analytical Chemistry. Firstly, look at the syllabus content. For this topic, you will learn types and steps in analysis, review the terms moles, molarity and concentration, other expression of concentration, percent, weight per weight, weight per volume, percent volume per volume, ppm, ppt, and ppb. And also you will learn the calculation involving different expression of concentration. Now look at the learning outcome. Upon completion of this topic, students should be able to differentiate between qualitative and quantitative analysis, explain the step involved in quantitative analysis, calculate the concentration of solution, and explain the preparation of solution. Firstly, look at what is analytical chemistry. Look at the definition for chemical analysis. So chemical analysis is the determination of chemical composition or chemical makeup as well as the quantity of each composition present in a sample. Example, if you are given with the blood samples. So for blood sample, the chemical analysis of the sample involve the determination of its iron content, its alcohol content, or perhaps its drug content and how much each of the composition presents in the sample. If the sample is water, okay, water sample usually in terms of river water, wastewater, effluents, or ocean or lake sample. Yeah. So the analysis of a water sample involves the determination of its mineral content, its pollutant content, and its dissolved oxygen content, and the percentage of each constituent. Importance of analytical chemistry. So what is the importance of doing analytical chemistry? So actually the analytical chemistry provides methods and tools to collect data to solve problems in field of science such as industry, medicine, environment, chemistry, biology, agriculture, material science, archaeology, forensic science, geology, physics, engineering, and so on. Okay, so in analysis, qualitative and quantitative information are required. Okay, look at the types and steps in analysis. Firstly, look at the discipline of analytical chemistry. So in analytical chemistry, there are divided into two analyses. One is qualitative analysis and the other one is quantitative analysis. What is qualitative analysis? Qualitative analysis is an analysis to identify the constituent present in a sample. What are the constituents? So it can be elements, atoms, ion, and compound. Okay, actually when you determine when you do qualitative analysis, actually you want to know what it is. Okay, so now look at the quantitative analysis. Quantitative analysis is the analysis to determine the relative amount of the constituent present in a sample. Okay, so in qualitative analysis, you want to know what are the compounds. In quantitative analysis, actually, you want to know how much of the compound. It is how much. Okay, look at into detail the difference between qualitative and quantitative analysis. Refers to this table. Qualitative analysis is to determine the presence or absence of a substance in a sample. So when you do qualitative analysis, you will determine the chemical identity of the substance of the samples. So you may determine the types of species, analyte, atoms, ions, molecule or functional group. Example of chemical identity, the presence of copper ion detected when flame when flame color is green, or identifying an ion and cation in aqueous solution. So using this referring to this example, so you know the presence of copper ion in the sample when we do flame test. The flame color is green. So you know the presence or the absence of the copper. Okay. Qualitative analysis achieved by, so referring to this actually, how you determine the presence or absence, what are the tests that you do in order to determine the chemical constituents. So it can be achieved by color changes. For example, you do flame test. Okay, if the color of the flame is green, so you know it is presence of copper. Or you might do distillation, extraction, or you might use gravimetry method using precipitation, or you use chromatography. It is actually separation method. Okay, referring to the quantitative analysis. Quantitative is to determine the exact amount of the substance or analyte in the sample. In another word, you want to know the how much is the compound in the sample. Okay, quantity of the species in a sample, such as concentration, level, percentage, mass, relative abundance, and so on. Example of quantitative, the concentration of oxygen in blood sample is equal to 3 ml oxygen per liter blood. So for this case, what are the compound? It is the oxygen. How much is the compound? 3 ml. Okay. And then another example, percentage by mass of silver is 42.88% in ore. So when we determine quantitative analysis, means that you will know how much or the quantity of the substance in the sample. So for this example, what are the substance? It is silver. How much is the substance present in the sample? 42.88%. Okay. How you do the test, or how do you determine the quantity or the amount of the substance in the sample? You will do the method or the analysis using absorption of light, density, chemical reaction, or precipitation. Or you might use volumetric analysis, which is when you do titration. What is analyte? So analyte is the chemical substance in a sample that are being identified. Okay, look at the method of chemical analysis. So to estimate the concentration of analyte may require both measurement of the mass or volume of the sample and some physical quantity that is related to the concentration of the element or compound. So this quantity can be classified as gravimetric, volumetric, electroanalytical, spectroscopic, and miscellaneous. Okay, look at the exercise. State the type of analysis. Either it is qualitative analysis, quantitative analysis, or both. Look at the first one. A, to determine and compare the percentage yield of fish in a siakap in siakap and tilapia fish. So is it qualitative, quantitative or both? Determine and compare percentage. So when you determine and compare the percentage yield, so it is actually the amount. So when amount, it is quantitative analysis. Look at B. To determine the presence of ascorbic acid in canned fruit juice. Presence of ascorbic acid. The keyword is here. So presence or absence. So it refers to the qualitative analysis. C. To determine the amount of potassium content in varieties of local durians. Determine the amount of potassium in varieties of local durian. So varieties local durian, there are many samples. So they want to know in the local durian, is there any presence or absence of potassium content? And also they want to know the amount. So for C, it is both. Okay. Qualitative and quantitative analysis. D, a hazardous waste disposal site is believed to be leaking contaminant into the local groundwater. Is there any amount determination? No. Dia cuma nak tahu is there any leaking contaminants. So it refers to the presence and or absence. So D refers to the qualitative analysis. E, a quick way is needed to find the level of blood pressure among men above 60 years old that can categorize as high BP. So they want to know the level of blood pressure among men above 60 years old. So it refers to the both analysis. And F, a reliable method is needed by airport security for detecting the presence of explosive material in luggage. The keyword is here, presence. So when you want to know only the presence of the substance, you do not want to know the quantity or you do not want to know the amount. So it is qualitative analysis. Okay, now look at the steps in analytical chemistry. So the first step, defining the problem, selecting the method, obtain a representative sample, prepare the sample for analysis, eliminating interferences, performing the measurements, and finally calculating the result and reporting the data. Look at the first one, defining the problem. Okay, look at define the problem. You will look at a few factors in order to define the problems. Actually, when you define the problem, in other words, you find the topic or you want to find what are you going to do for this research. Okay, so factors to be considered when you define the problem is what is the problem? What need to be found? Qualitative and or quantitative. What will the information be used for? Who will use it? Okay. And then when will it be needed? How accurate and precise does it have to be? What is the budget? The analyst or the problem solver should consult with the clients to plan a useful and efficient analysis, including how to obtain a useful sample. Let's say you already define what is the problem, but you have to know the budget. So if you want to solve some problem that the sample may be on the moon, okay, you get the sample from the moon. So you must look at the budget. Can you do the research? Do you have enough budget? That's why you must consider all these factors before you define the problems. Once you have defined the problem, now you have to look at select a method. So what type of method to be used in order to do the research or in order to solve the problem that you have identified. Okay, for selecting method, you must consider certain factors. The factors are sample type, size of samples, sample preparation needed, concentration and range, sensitivity needed, yeah, selectivity, accuracy and precision, tools and instrument available, expertise or experience, cost, speed, does it need to be automated, method available in the chemical literature or standard method available. So when you do research, when you um, consider to do some research, so you must select the method which is available in your organization. For example, you want to do research on apa ya? Purification. For example, you want to do isolation and you want to purify the component, and then you want to identify what are the components in the sample. So you need to do extraction, you need to do isolation or purification. So you might use chromatography, you might use radial chromatography. So you have to consider, do we have radial chromatography in our lab? Okay, or you want to identify the pure compound that you have purified. We use NMR. Do we have NMR in our lab? So if we do not have the instrument or we do not have the chemical reagent needed, so you must consider the sample, or you must consider doing other analysis, or you must consider to choose another method. And then obtain a representative sample. So you must consider sample type, homogeneity of size, and sampling statistic and or errors. And then prepare the sample for analysis. You must consider the factors. Is it the sample in terms of solid, liquid, or gas? So will it dissolve? Can it use ash or digest? Chemical separation or masking of interferences needed. Need to concentrate the analyte, need to change the analyte for detection, or need to adjust solution condition. For example, you have to adjust the pH or you have to add reagent to the sample. So when you have to consider, yeah, and then perform any necessary chemical separation. So let's say your sample extract from the plant, okay, the crude extract. Let's say you want to determine only the tannin, or you want to extract only tannin. So when you extract the sample from the plant, you will extract all the compound from the plant. If you want to do analysis only on tannin, then you have to remove other compound. So you might use a certain method to remove other compound that you don't want. So you may use distillation, precipitation, or you will use solvent extraction. You have to choose a specific solvent to extract the compound, or you might use solid phase extraction, or you may use chromatography, or you may use electrophoresis. So this may be done as part of the measurement steps. And next step is to perform the measurement. So in order to perform measurement, you must consider the factors, calibration, validation, control, blanks, and replicates. And last one, calculate the result and report. So you will do statistical analysis or reliability test, or report result with limitation or accuracy information. Okay, now look at the defining the problem, step 1. Just now we discussed the factors to be considered. Okay, now look at the defining the problem, step 1. So what should you do? So in order to defining the problem, actually it is to obtain required information within a period of time. So you know what information is needed, by whom, what purpose, and what type of sample is to be analyzed. So by now you must know where to collect your sample, who will provide the sample for you, how will you do the sample collection. So all of this defining the problem, it depends on equipment available, cost and time involved. Step number 2 is select method. Yes, now we discuss the factors to be considered. Okay, so for select a method, you must know origin of the sample, factors to select a method, which is sample type, sample size, level accuracy, sample preparation needed, or transportation or storage of sample. Okay, so you must know how much sample you need to collect. For example, you want to do research on plant. Contoh stem bark. Okay, let's say the sample is stem bark, sample type. So sample size or size of sample. So how much or what is the amount of the raw material you need to collect? So let's say for your analysis you need 1 gram, for example. So you must collect raw material about 4 to 5 kilogram. So because through the process, you will cut it, you will dry it, you will grind until it become powder. So once it become powder, you have to do extraction. After extraction, then you can get your crude extract. The crude must be 1 gram. So you must know the percent yield. For example, if you want to get 1 gram, how much is the sample? Or what is the mass of sample you must collect? It might be more than 1 kg. Okay, that's the example. Okay, so complexity of sample and number of components in sample influence the choice of method applied. And look at number 3 or step number 3, obtain a representative sample. So representative sample, a small quantity or portion of material that represent the whole sample of population. So you must know the type of your sample. Is it solid, liquid or gas? Sample may be homogeneous or heterogeneous. The suitable sampling method the suitable sampling method differ from one substance to another depending on homogeneity. Gross sample consists of a portion of tested material, so must be reduced in size to gain laboratory sample. So lab sample, so it is small portion crushing or sieving to form homogeneous or uniform powder for analysis. So the analysis sample that actually analyze. So step number 4, prepare the sample for analysis. So actually this is preparing the lab sample. Solid, grinding, mixing, storage to avoid changes. If the liquid, solvent and analyte evaporation, storage to minimize chemical changes, you will use refrigeration, freezing, deoxygenate, or you must put your sample in a dark place. Preparing and storage the solution, physical and chemical changes can occur, no suitable solvent use, acid, base, oxidant, reductant or organic solvent. Step 5, eliminating interferences. Perform necessary chemical separation to remove interference. Impurities may decrease the spectroscopic signal used to calculate the concentration. Separation steps include precipitation, extraction, chromatography, or distillation. So, you must know what is interference. So, interference is the species other than analyte that can cause error and affect the final measurement. For example, your sample is leaf from the plant. So, when you do extraction, the crude extract obtained may be green in color because it is leaf. So the green color actually it comes from chlorophyll. So you don't want chlorophyll in your sample. So it means that the chlorophyll is the interference. So you have to remove the chlorophyll first before you can do further analysis. So you might check what are the methods to remove chlorophyll. You might use distillation, you might use liquid extraction, or you may use other reagent to remove the chlorophyll. If reagent, maybe we use charcoal, okay? Or if you do chromatography, we might use Spadex. Step 6, performing the measurement. So for performing measurement, you must replicate your sample. So it will improve the quality of the result and provide a measure reliability. So for analysis, incorporate the measurement of the concentration of the analyte in replicates and comparing with standard. So you might do gravimetric analysis and volumetric analysis. For gravimetric analysis, it will be based on measurement of mass. You might do precipitation. Or for volumetric analysis, it based on measurement of volume, which you can do titration. So in step number 6 also, performing the measurement, you might use instrumental technique, more selective and sensitive than volumetric and gravimetric analysis. So for this case, you might use instrument. You might use HPLC, AAS, and so on. Okay, instrumentation must be calibrated. So calibration is accomplished by preparing a series of standard solution of the analyte at known concentration. For example, this is the standard. So you run with known concentration, and then when you want to determine your concentration of your sample, you might use this calibration curve. So calibration curve, instrument response function of concentration. Step number 7, calculating the result and reporting the data. So for this step, you will calculate and evaluate the result to estimate whether the results are reliable or not. You will do precision and accuracy. Precision is estimate using the standard deviation based on replicate measurement, and accuracy evaluate by referring to the standard material. Precision and accuracy is very important in analytical chemistry and referred as quality control or quality assurance. So detail on precision and accuracy calculation we will learn on chapter 2. So that's all. Thank you.